Good morning to you all. I'm happy to talk for you all and to see that so much interest. What I will share with you today is what you see here, nutrition research in aquatic environments. But what I especially want to show is how you can start from making a concept for research to get it finally into a publication. Because I can imagine that for some of you that might be new, and I would like to go with you through the different steps of what you could say is the science business. So we can start with the project proposal, of course. When you make a project proposal, whether it's on aquatic nutrition or another topic, of course, you should first do a literature review. With that literature review, you can then identify the gaps, what is still not known, what should be known in research. And then, and this is not a typographic error, you do a literature review again, because once you have identified what is the gap, you want to know more about that specific thing that you would like to investigate. And once you have done that, you can formulate a hypothesis or an aim. And not every research is hypothesis driven, so it can also be an aim that you want to explore something new, and from that you make a study design. So a literature review, why should you exactly do that? Because you want to avoid repetition and unnecessary studies. If you do a lot of effort to study something and afterwards you find out that somebody else has already done it, then it will be very difficult to still publish that in a good journal, because most journals will not accept, will go to reviewers, and if reviewers see like, okay, this has already been done, then they will not accept your manuscript in the journal. But also you need to do it to avoid wrong conclusions based on a subset of data. Sometimes when you have incomplete information, you may think like, okay, this is the rule, but when you look at the entire data, maybe something else would come up. Sorry. So how can you do a literature review? You can do it in different ways. You can, for instance, make a systematic review. This means that you will dig into literature and based on specific criteria, on specific keywords, you start looking for a set of papers and you define the set that you actually are going to analyze. Or you can do a meta-analysis. A meta-analysis is a lot of work because it's based on the crude data that you have to gather from different authors. So not just the published data, but really the figures that they used to make their analysis with. But based on that, you can have a very good idea of what happens in literature. So it's not an easy task, but it needs to be done and is essential before you can start a good research. When you come up with research plans, it is good to have one specific research idea, and not 2, 3, or 4, but that you can define it into one specific research idea. I give here one example. You could say, with the relevance of the question, are aquatic organisms such as amphipods with high natural UV exposure, like in Lake Titicaca, are they at risk of vitamin D deficiency when you keep them ex situ, when you start breeding them in other places than at high UV exposure? This bank can be quite relevant because if you want to promote the conservation of a certain species, you need to know what are the factors that can affect its life. Now, this is an example. So what you then need to do is start looking what is published about this topic. And without being complete, I mentioned certain things. So keep that idea in mind. We want to see, does UV exposure matter for those organisms? So you want to know, how do organisms adapt to high UV exposure? Is there already something published both on behavior, but maybe also on the physiology, so that the body can adapt to high UV exposure? You also want to know, because UV is very important for vitamin D synthesis, how does vitamin D deficiency develop in low UV environments. We're talking about aquatic organisms, so you also want to know, is it really an important issue? Because we know UV, the UV strength decreases very fast when you go deeper in the water. So for which organisms is this an item actually? And of course, you want to know, are there other aquatic sources of vitamin D for those animals or for other animals? And other topics. So you need to cover really all the different aspects so that if you come up with a proposal, something you might have to submit to a government or to a funding organism, that at least you know this is new and this is relevant. So you need to check for novelty. What does novelty mean? Novelty means that you have to ask yourself, did other researchers already publish comparable studies? I will give some examples here below, but it's not a problem if you find something on that topic that you say, oh, I should skip this item because it's already been done. No, if that's the case, did they provide the proper answers to the research question? And I give this as in an example. Suppose that you find that a research group already described the relationship between altitude above sea level and vitamin D content in amphipods. That's certainly an interesting piece of work for you in your preparation. But it doesn't mean that it already solved the question, because there are several reasons to say why it's still very relevant for you to continue the research. Finding a relationship does not mean it's a causality. So the association between altitude and vitamin D may not be causal. It may be that altitude or UV exposure is not the real reason for the difference in vitamin D level. Maybe the lower vitamin D level is just reflecting different requirements at lower altitude. And then you're looking at a different thing. And it would be very interesting to figure that out, to see, is it a causal relationship or not? The association may does not be because of UV. And also the difference in vitamin D may actually not be a problem for the animals. And that's coming back to something, the next step. But here you also need to check that, because if people say, oh, you need to take that into account for the vitamin D status of the animals, then maybe your task would be to see, is it really a problem? Because for some animals, it may not be a problem to have lower vitamin D if it's still sufficient. So that is a case where you can still say, okay, there's already some work being done, but there's plenty of opportunities to further continue on that, to still have novelty. It becomes a little bit different when you find that the group already has studied that amphipods from a high altitude lake kept at low UV exposure developed actually signs of vitamin D deficiency, whereas compared with amphipods from a low altitude lake are not developing that. Because that's actually a proof that you need to take into account the vitamin D supply by UV exposure for those high altitude amphipods. So you could say this is proven. Is there any opportunity to further work on that? Well, not so much anymore, except it can still be interesting because maybe that group didn't look at all at possible mechanisms. So it might still be very interesting to see what is the evolutionary mechanism against high UV that caused the vitamin D deficiency at low UV exposure. In other words, if you can find out why those animals are actually developing vitamin D deficiency, it can help in general, and not just for these amphipods, but in general, it can help understand how you have to deal with vitamin D deficiency in those cases. And then it's still relevant to go further into that. But that's to check the novelty and find the gap in science that is still there. So it's not only important to check the novelty, as important is to check whether your study idea has relevance. In other words, will the scientific world recognize your idea as something that matters? Do people think what you're doing is something important? I give a bit of a funny example, but you could say, we are the first to investigate the effect of alcohol percentage in Belgian beer, on the survival of amphibots in maybe Bolivian peanut soup. I'm sure nobody else has done that before, but so it's very novel, but nobody will care. You will not get funding for that because it's not recognized as something relevant. And this is now a bit of a funny example, but you always have to think that something you think can be important for yourself, if you expose it to the scientific world, you should always think like, is this something that is of relevance for the scientific community as such internationally? And then finally, you have to check for novelty, you have to check for relevance, you also have to check for the expertise. You should not assume that you know everything yourself. I do that always for every project I do. I always think like, okay, what type of disciplines do I need to have a full coverage of the expertise that I need in a certain project for a certain idea. So question is, do you have the necessary expertise to investigate such a topic? If not, that's not a problem. Then you have to think for yourself, can you train yourself or look for collaboration? Again, an example, suppose you want to evaluate if vitamin D deficiency is developing in Hyalella amphipods when exposed to low UV. But you don't have the equipment to measure vitamin D, for instance. You don't know how to take and conserve the samples because you haven't been studying vitamin D yet, and you have little experience in the topic as such. Should you then stop by doing the study? No. Dare to ask for collaboration to experts and negotiate under which conditions. Sometimes they will say, we can train you to do these analysis. Come to our lab and we will show you how to do it. Or we send you an instruction and we teach you how to do it. But it can also be that they will just say, okay, send us the samples and we'll do it for you. The reason why I say dare to ask for collaboration is because sometimes you might think like, oh, we shouldn't ask these very big, very known researchers to ask for collaboration, because sometimes you do have something interesting to offer for them. Not everybody has access to high altitude lake. Not everyone has the expertise to study fish or frogs or invertebrates in the lake. So sometimes you don't have to be scared to ask for collaboration. We always say, you have a no, you can get a yes. And that's my call. Never be afraid to ask for collaboration. Usually people can say yes one way or another. So when you check for expertise, and this is just one of the examples, this is from one of my former Ethiopian PhD students, a paper on the exposure of water temperature effects in fish. And I just want to show what co-authors are about. So first, of course, you have the first author, which is the Ethiopian PhD student that did the study. The second author there is an expert in fish morphology that we involved because we said, okay, we don't know all the things about how to dissect the fish, what exactly to take out and how to preserve, etc. So we involved her. She's from another department of our university. And then we went to Norway. Why? Because these people that are indicated here are experts in amino acid metabolism in fish. And that's what we wanted to study. We knew something about amino acids, but they could analyze them and they could help with the interpretation. So that's the thing. And then there's me and Marta, one of my former collaborators. We have expertise in animal nutrition. We were also supervising the Ethiopian student. So you see, we have Belgium, we have Norway, we have Ethiopia all involved just because we have been looking around the globe who can help us with this study. And this is not an exception. This is a standard for most of the studies we do. So then we come to experimental design. Once you know what you want to do, you have to start thinking like, how will the study look like that I want to do? Whether it's an experiment in a lab or whether it's a field study, it doesn't matter. It's always important to think how you will proceed. So first you need a clear research question, not like we have the opportunities to start sampling, let's start doing this. No, you have to get one idea. And why is that so important? Because if you don't have a clear idea, you have no clue what to look for, how to preserve samples. I'm just saying, if you catch fish from the lake and you don't know what you will analyze, you don't know, do you need to freeze them? Do you need to store them in alcohol, in formaldehyde? Do you need to do something else? Because afterwards, if you then only decide what to do, maybe you didn't properly. So formulation of your aim or your hypothesis. What's the difference between an aim and a hypothesis? An aim is mainly meant when you do exploratory research. You could say what Charles Darwin did when he discovered the theory of evolution is exploratory research. So it can be very valuable. It's just going out and say, okay, we aim, you have a clear research question. In the case of Charles Darwin, he was indeed looking like, why are animals becoming so different? Where does this come from? So that's a clear research question, which made that when he started selecting his samples, he was already looking for something. A hypothesis is when you say, okay, we already have kind of information and we want to test something very specific, which is an explanatory research. You see something, you have already observed something, and you want to know how it works. So you make a hypothesis, you say, okay, we think that it works like this. For instance, we think that Hyalela from Titicaca Lake, when we put it away from UV, that it will develop vitamin D deficiency. That's a hypothesis. And a hypothesis is always a hypothesis that needs to be evaluated. So it can be right, it can be wrong. And the wrong can be as valuable as right. So then you come to the study design, and then automatically you end up with statistics. Before you start the study, you should actually already know the statistical model you are going to use to analyze the data. And why is that? Because if you just start sampling around, maybe afterwards you run into troubles to get it analyzed statistically. You have lots of samples, but maybe no idea of how to analyze it. And then you end up with nothing. So it's very important to think about your model, statistical model, which groups will you compare, which parameters will you link with each other, that you have that done beforehand. If you're not acquainted to do that, just ask help from people you might know that have some background in statistics. And then, of course, think which analysis need to be done. Don't try to measure everything. I do a lot of reviews for several journals, and what I very often encounter is that people have a machine in their lab, for instance, for blood analysis or something, and whatever they are investigating, they put the blood samples in that machine. And then my question is, okay, what's the relevance? Why would you, for instance, just to say something, measure glucose in the blood of a fish? If your hypothesis has nothing, or your aim has nothing to do with that. So when you think about parameters, again, think globally. Just don't think like this is the only thing we have, this is the only thing we can analyze. Think like what should be analyzed in order to evaluate your hypothesis or to obtain your aim. And this can be something which is not at your disposal at the moment, so therefore you have to look for other expertise, maybe other labs that do have it, and ask for collaboration. So that's very important. In order to get progress, you start from the idea. From that, you see what you need. And then final bit within this part is to make the difference between a short-term and a long-term deliverable. What I mean with this, do not try to answer a very big research question with one experiment or one study and one field study or something. The reason is we, with our human brain, we cannot contain all these different parameters. And you often have too many parameters. You lose the picture of what is really happening, and you lose track of the question. There will be lots of things coming in, and sometimes it's just better. You can always store samples if something interesting would come up afterwards, but always start with your clear research question and try to analyze those parameters that you think are needed to evaluate it. Not more, not less. Think very carefully. I see that that problem occurring a lot, that people try to solve a very big question in one study and then fail because they try to do too much at once. So you better do a kind of a set of smaller experiments which can have short-term deliverables. Each one can be a piece of a publication, but in the end, when you take all experiments together, this is the answer to the big question. And that's typically what people do in a PhD study. You have several studies, each with short-term deliverables, and at the end, you take it all together and do a kind of an overall analysis like, okay, where are we now in the big question? But this is not only for PhD students, this is also as a researcher, think long-term. So think, okay, which steps, which small experiments, which smaller studies do I need to make progress so that in the end you can answer the big question and maybe even get a big paper out of that when you compile everything. So that's then your long-term deliverable. So that's the deliverables, and then how to develop the research proposal. You have to analyze the specific scientific problem. In this case, what are possible mechanisms to cope with high UV exposure? If you want to do mechanistic research and find out, okay, we see that something is happening with this high UV exposure, how do the animals manage to survive? So that might be different. I gave you 3 options, but it doesn't mean that those 3 options are automatically the only ones. It's just the 3 that I thought of first. First, you could say, okay, maybe there's just behavioral response. Animals hiding under rocks when UV exposure is too high. At midday sun, and you indeed can see that some of the animals in the lake will just hide under the rock to be protected from sun and UV. You can study that and it can still be interesting, but when you think about, okay, how does it then come that they would develop vitamin D deficiency when they're devoid of UV, then maybe that's not a solution. Because if you think logically, if hiding would be the only adaptation, then they should not develop vitamin D deficiency when they are not exposed to UV, because hiding means they are not exposed to UV. So a further possibility might be, okay, if the animals can produce lots of UV, lots of vitamin D, they might have a higher calcification of their exoskeleton. When you talk, for instance, about Hialela. To evaluate this, evolutionary solution would make sense in terms of costs. So you need to evaluate that. Does it make sense that an animal would do that? Is it beneficial to have a harder exoskeleton, or rather a burden? Because it's not because an animal produces something that it can produce something at a higher level, like calcium, building blocks, that is beneficial. Maybe it's just a burden, and therefore it would not be a good evolutionary solution. The other thing is UV exposure is also about DNA damage. So just looking at vitamin D in the physiology of the fish would not be sufficient. Something that could be interesting is to look at pigmentation. We know that individuals, and that's the same in humans, humans that are living at higher UV exposure typically have a darker skin. I don't live at high UV exposure, you can clearly see that. But then you have to think what is known from other studies. What can you already learn that can bring you further ideas? And then sometimes the most simple things have not been evaluated, like to which extent does Hyalela at a high altitude lake differ in pigmentation from a low altitude lake? This can already bring you to some ideas. Are we looking in the right direction to find mechanisms that are coping, that helping coping with high UV? So of course this is not complete, but I just want to give you the idea of how you try to analyze the problem. And this is still before you have done one research effort. This is still in the phase of making the idea, and just need enough time to develop that. Okay, once you have done all the work, I'm making a big step now, already past having done the work. There's two important things that you need to consider: the quality control of your raw data. So meaning, be critical. Don't accept that everything you find is really true. Always evaluate, haven't there been any errors in the machines, in human errors that have occurred? Have you been biased? There's lots of studies in literature that show that unwillingly researchers can be biased, because you want to have a certain result, you have a hypothesis, and you would very much like it to be true, and therefore you might unwillingly deselect some of the data or not look into certain data. So it's very important to be critical, because sometimes finding something which is contradicting your hypothesis can be the most interesting one. If you find something that doesn't make sense at all, but you find it all the time again, then you might bump into something really important. So don't accept everything as true, be critical. Always think about replicates, averages, standard deviation, coefficient of variation. So the data variability within replicates. So you have to explore and explore to see, to learn to know your own data. Make a lot of plots, graphical designs, so that you really see, okay, how are my data moving? What do you do? Even before you do any statistics, that you have an idea of what you're looking at. Was your method okay? Have there been any human errors? Would you be able to repeat? Are there any particular outliers? If you suddenly see that a value is 10 times higher than it should be, then you have to think very critically, where did it come from? Before you throw it out, always think should I throw it out? Maybe that individual if a fish has, let's say, or if Hyalela has 10 times more pigments than another one, maybe that's a very interesting observation, and you don't want to throw it out, because actually, although it's an outlier, it might be a very meaningful outlier. When you look at populations, then you can say, okay, let's leave this outlier out, and we're just looking at averages within a population. But treat outliers very carefully. Think critically, should we throw them out or not for the purpose of our study? And then we smoothly come into the statistical analysis. You have to think, what is your experimental unit? Even if you don't do an experiment, you have to think what can you independently analyze? And that's not always as easy to identify. So which type of model are you using? And I give this a very simple example. This is more for an experiment. When you would put, this, you could say these are fish or this is something else. Actually, it doesn't matter. You have a bunch of individuals in one tank and in another tank. This is and the left tank one is your control group. And on the right, you have your treatment group. So in our case, and the example that we're working with, you could say the control group is getting UV and the treatment group is not getting UV exposure. All animals within a tank are treated individually. You measure them individually. So the question is, how much independent replicates do you have? If you now start counting the number of fish in every tank, then you're wrong, because the number of replicates is actually 1. With such a setup, you can never do proper statistics, because you don't have independent replicates. Meaning, if something happens to tank 1, for instance, not only the UV exposure is different, but maybe there's some pathogen, some disease coming in, or accidentally something put the chemical in there, and your animals are doing worse. Then you would say, oh, it's because the trees compare statistically. If then there's one tank where a disease outbreak is in, you can still throw it out, and you will clearly see that it's not because of the treatment. This is, of course, a bit more difficult when you start comparing, for instance, regions within a lake, because you could say a lake is one big tank. Of course, in that type of research, ecological research, it's very important, therefore, to describe why you're not looking at one tank, why your observations are still independent, because the conditions where you measure are different. That's the reason why in ecological research, it's much more important to describe location, circumstances, water quality, etc., to assure that what you're comparing are different circumstances. So then the interpretation of the data. Always be critical. This is not just a vain word. With be critical, I mean, again, if you have analyzed the data truly and you see differences, always think like, okay, can there be a reason why this is occurring? And try to discern what you find as statistically significant difference versus biologically significant difference. Maybe, just to give an example, if you look at the vitamin D level in the Hyalella within your experiment, you find a 1% difference in vitamin D level. Maybe it's statistically significant, but the 1% difference will not mean anything to the animals. There will not be more or less disease because of that 1% difference. So you can indeed say we find something statistically different, and it can be important to report that. But when you start interpreting, you should not say, oh, this group had a better status of vitamin D, because that's not the case. It was only a 1% difference. So drawing conclusions is conclude only what data allows you to. Sometimes people start building on their data and say, okay, when we see a 1% difference in vitamin D, it means that the group with the lowest vitamin D level is less healthy. No, you can't conclude that because you didn't measure the health of the individuals. So you have to stick to your data and say, it may mean something to the health of the animals, but we don't know. We can just report that there is a difference in vitamin D. So this is an example of this. This is not about aquatic animals. It's just an example to show. This is a study with adding tannins to the diet or not. And you can clearly see there's two batches, batch 1 and batch 2. So the two left ones are batch 1, the two right ones are batch 2. And you see that in both cases, the tannins lead to a lower protein digestibility. This is quite well known that for a lot of tannins, when you add that to the diet, protein digestibility goes down. But when we talk about the relevance, the importance of that effect, you see that comparing one batch with another, and batch meaning also another type of diet, that the diet itself, that the batch has a much bigger effect than the tannins themselves. You can see that when you compare batch 1 with batch 2, that batch 2 is much, much lower, and that effect is more important than the one of the tannins. So tannins do have an effect, but when you put it in the larger context, they're not the most important effect. And that's how you should interpret the data. Don't automatically think that what you're studying is the most important. That will be the strongest paper if you are modest about that. Okay, so once you have done all the work, you have your data analyzed, then we start about writing a scientific paper. And writing a scientific paper, I put here 3 items. First, you need to say, okay, you check for a journal, and every journal has author guidelines, meaning every journal gives you a set of rules that you have to adhere to when you write a paper. It's very important to check that in every detail, because you can annoy a reviewer or an editor when you are not conforming to those journal guidelines. And some editors will just reject your paper without even having read it. So then we come to the sections of the scientific manuscript and something about the ethics or the deontology. Choosing a journal to publish in. One check I would advise is check Beall's list of predatory journals. I'm not sure if you're acquainted with the term predatory journal. A predatory journal is actually a journal that has not a really sincere aim of publishing science, but just want to make money. There's a lot of journals where you have to pay for now to publish, and you can discuss about that. But there's a lot of good journals that do that and just try to cover costs. There's also journals that will readily accept your data, do very poor reviews, so it's very easy to publish there, and then they will charge you to put your paper in there. So these are the predatory journals, and it's not always complete, but at this website of the Beall's list, you can check whether a journal can be trusted or not. So if you want to avoid that, preferably choose from the JCR list, which is the Journal Citation Reports, or ICR is an abbreviation you can look for. So these are journals with a JCR impact factor. Familiar with the term impact factor? An impact factor is actually a kind of a number that a journal gets, and this is calculated based on how often a journal is referred to, an article is referred to in the past 2 years. Just to give you an example, if you publish an article and your article if you publish an article today in 2022, and you look in 2024 and you see that your paper has been referred to 3 times, then your paper has an impact factor of 3. So you understand that the higher the impact factor is a kind of an assessment of how important something is, how important a journal is, how important an article is. Of course, it's not the only thing that should count. In some disciplines, it's much easier to get a very high impact factor than in other disciplines, but it says something about the importance. And as soon as it has an impact factor, you can assume that it's a trusted journal, even if the impact factor is low. Now, Journal Citation Reports, the problem is you need a login for that website, and it's quite costly, so not all universities have that. So in that case, I just would suggest ask a colleague from another university that has access to JCR to ask for the impact factor of a journal, whether a journal has an impact factor or not. Sometimes you can find it online when you type the title, for instance, Journal of Ecology, and you put their impact factor, you will typically get whether it has an impact factor or not. But it's good to check that. So publishing in predatory journals can be very easy, but when you want to have international collaboration or even want to move internationally, it's typically considered as a weak element on your CV. You might think, I have lots of publication, but people will judge that as a negative point rather than a positive point. So it's good to check that. Okay, so how do you build a paper? You start with an introduction, and the introduction should start with what should it have, the rationale and the relevance of studying the topic. So why do this? Yeah? What is known? So meaning, what did you get out of the literature review? How did you identify the gap? What is still not known? And then from that, you formulate your hypothesis or your aim with the specific objectives of the study. I made this on one of our papers. This is again a study on fish Coincidentally, it's again from an Ethiopian PhD student. So I'm not going to read it all, but just go through the different steps. So I show you the introduction of this paper. What we do in the first part is describing what is a scientific problem. Here it is like it is not well known how the available diet of fish affects their development. Studying in different lakes, in different situations, how important is nutrition for the development of fish in different environments. And then, of course, that's a scientific problem. Why is that still a scientific problem? Because the gap in science, why is that gap still there? Is there are studies on fish nutrition, but this is usually in lab conditions, not in natural conditions, to clearly mimic the living environment of the fish. So it's better to study fish nutrition in the wild, but that's of course difficult because you cannot exactly estimate what they are eating. In the meantime, there's some solutions for that, but if you want to know what does it do to the fish, then there's the opportunity. And that's where we come in with this piece of text describing, okay, what is our novelty? What did we try to do to fill that gap? Is we have thought of a method to study fish nutrition in the wild. And we describe how that method is, that it's already validated for other things so that we don't start from scratch, but it's still a novelty. And then finally, from that, we built our aim. We say, what is there for the aim of the study? Meaning we used our method to show that fish nutrition can be studied in the wild. And for that purpose, we have studied Nile tilapia in Ethiopian lakes. And why I put the next sentence, the last sentence, in capital letters is because I think that's something very important. We say, okay, we study Nile tilapia in Ethiopian lakes, but that is just a suitable model to validate our study wider than that. Often the problem is that you do a study in a certain condition, and then you say, okay, we study, for instance, the population of Hyalela in Lake Titicaca. You send it to an international journal, and the typical reply will be, this is not for our readership because this is a very local thing. It's not adding to general insights in science. So always think like, okay, how do our study, how does it bring addition to science in general? Not just for that particular species in that particular location, but broader insights, mechanisms, insights that you have in nutrition, physiology, biology that you can mention. So then material and methods. So we've had the introduction, material and methods. With material and methods, you have to describe everything that allows somebody else to repeat your study. So you carefully have to think like, what are all the details that need to be in there? Some of the typical things, but it depends on the type of study you do, whether you have to describe. Of course, you have to describe your species. If you're working with a fish species, for instance, Orestias or something else, you need to describe, okay, this species, what is it about? If necessary, you have to mention whether your study had been evaluated by an ethical committee, meaning if you did something with the animals that can harm their welfare, then more and more journals will just ask whether it has been approved by an ethical committee. You need to describe all circumstantial data, environment, location, available diets, whatever is important, like I say, to replicate the study that you did. The study design, what were your groups that you were looking at? How much samples did you do? What was your experimental unit? And the repetitions you made. And then, of course, the methods used for each parameter. For some parameters, which are quite common, you can refer to a reference without explaining too much. When you have something novel that you not a lot of people have been using for that purpose, you need to describe that method and sometimes even explain that it has been validated for that purpose. Just a very simple thing. If you want to measure glucose in a fish, but the glucose method has never been used for fish, then you might need to add a small part that shows that it works, and that it validates that it's reliable. You need to mention how certain calculations are done, and then of course the statistical methods. And also just measure all things that you did when you have your raw data. What did you throw out and why? Were there certain animals sick? Why you didn't involve them? Were there certain errors in measurements that you know of, why you had to kick them out? But you need to explain so that reviewers afterwards do not find odd things and start doubting about the value of your study. So report what the data shows you, not more, not less. Focus on data that is important for the discussion. Sometimes you have plenty of data, and it is allowed to, let's say, guide the reader towards the interpretation that you want to give. So you can make, if you measure, let's say, to give a simple example, if you measure 3 parameters, only 3, and you want to relate them to each other, then you can have 6 combinations of making graphs. But there you have to choose which one is for you, the one that fits into your aim or hypothesis, the one you want to show. So don't show everything. Try to guide the reader towards the story you want to tell. When you talk about tables, the captions, so the text that belongs to the table should allow the table, and that's the same for a graph, to be interpreted without the text. So if somebody only looks at your table or your graph without having read your paper, they should still be able to understand what it is about, and that's quite important. Titles of the columns and rows should be clear. Explain abbreviations every time you show something new. Don't forget the units that you're expressing. And then when it comes to tables, don't forget to think about significant numbers. I mean, I give here the example, if the standard deviation of what you measure is already higher than 1 unit, then there's no use of providing more figures behind the dots, behind the comma, because it's not meaningful. Then it's better also for readability to reduce that, and actually more correct to do that. Of course, p-value, SEM, sources of variation should be shown. A little bit the same for figures. So it should be standalone. You look, think about the units, the axis. Think about using colors. It's very easy to use colors now, but always think like, okay, if somebody would copy that on black and white, is it still readable? So you can use colors, but if that's the case, be sure that if you printed black and white, that your colors are still distinguishable. And make sure that the symbols are clear, which symbol belongs to which group. Okay, and then we come to a very important part, of course, which is the discussion and conclusion. In the discussion, what a lot of people do is start comparing with literature, like we found this and others have found that. We found this, whereas others have found this. This is not a discussion. A discussion is really getting the helicopter view, meaning you hang over your results, you know what is published in literature, and then you start looking, okay, you have some comparisons with literature, some things are different, why is that? Start discussing, build further on your hypothesis, but stay with your feet on the ground, don't become speculative, meaning if you build further on a hypothesis, you can only build when you have the bricks, when you have the stones. And the stones are your data or is literature. If you start thinking about wild ideas that you cannot support by data, your own data or published data, then stay away from it. Then it becomes speculation. So the conclusion, stay true to your data. Do not make conclusions that are not supported by your data, and come up with a very clear take-home message. What do you want people to remember from your study? And that's actually typically the last sentence of the discussion. Sometimes a separate part of conclusion, sometimes it's just the last sentence of the discussion. And then, not the most important part, but still something that people are struggling with, sometimes acknowledgements. You have to mention where did you get the funding from, the grants. but also people that contributed to the study. So not everyone should be a co-author. A technician is typically not a co-author. Somebody who helped with revising, and it can be very interesting to mention that. For instance, if you're not a native English speaker, it can be interesting to mention, like, this person has helped with improving the manuscript, the English language in the manuscript. Because that automatically shows to a reviewer, oh, they have done this review, so that's good. We don't have to comment about that because this has been checked. Also, free supply of materials, etc. This can all be in the acknowledgements. And then also a very important thing, the deontology of the ethics of publishing. I guess most of you will know about that, but I still like to put that forward. One point is never submit the same study to different journals at the same time. You can submit to another journal. So if you submit to a journal, the journal says reject or it doesn't fit within the scope, then you have all right to submit it to another journal. And sometimes a third, a fourth, a fifth, until it works. But never at the same time, because then you might have the chance that both journals are accepting it, and then you have self-plagiarism. And this is something that is considered as a very bad in the scientific world. So never submit the same study to different journals at the same time. Also, and I put it in bold, all co-authors that you involve on your paper should have read the manuscript before submission, or at least haven't given this consent, have agreed that you are sending it to the journal, that you are sending it for publication. I have had the situation that people have put my name on a journal that I was really not happy about, especially when it's about predatory journals. I am co-author of some papers in predatory journals that I didn't know of that my name was on. I've written to the journalist, but of course it's too late then. And let's say I'm quite sure that the people that put me on that paper, I will never work again with them. So be aware that co-authors should always be informed. All co-authors have to give consent to publish. Co-authors, who is actually a co-author? There's always some discussion, but basically it's about scientific contribution. It's not because somebody helped you to catch fish from the water that this should be a co-author. It's only when there is a scientific contribution. Somebody who thought of the concept, somebody who had a particular insight, who had a very specific technique that nobody else can do, those are people that you can add as co-author. It can be a technician. It can be a technician if a technician thought of a really breakthrough method to do something with a particular method, then yeah, a technician can be part of it, of course. So it's not that somebody should be staff of a university. No, everybody could be co-author if they had a scientific contribution. Conflict of interest is also a lot asked, and a lot of people automatically say, no, we have no conflict of interest. Sometimes you have. If it's clear that you work for an organization that is in favor of conservation, then you should be aware that and is against pollution, that blaming somebody for pollution might be an easy thing to do. So it's always good to be very honest about that. Also, may not be the case here, but if you collaborate with industry, you evaluate something that industry is producing, then obviously there's a conflict of interest, because that company that you work for has all advantage of finding positive results of the products they are producing. You have to mention that. If you're honest about that, most journals will not mind and will say, okay, they are aware of that and describe it honestly. You can also appoint preferred reviewers. This is making the life of editors easy, because editors have to find good reviewers. And when you work in a very specific field, the editor may not know the people that may be able to properly judge your manuscript. So you can help them. But of course, an editor will not use a preferred reviewer if it's obviously that you have already published 20 papers with that person, or whether it's your neighbor or your colleague. It should be somebody independent, somebody you say, okay, this is an expert in the field. I have no very close connection, so I can propose this reviewer. And then also a thing, you can send a cover letter to the, when you submit a paper. In a lot of cases, you can add a cover letter. And that cover letter is not just a vague thing. It can be important to explain the novelty and the relevance of your study to the editor. I give an example. This could be a type of cover letter where you say, dear editor, we hereby submit our manuscript. We think that our research is interesting because others have done it as well. We therefore hope you will publish it. Kind regards. This is not the way to do it, because basically you say, others have already done this, so we're not novel, and we're not pointing out what we add to science. So an editor will say, okay, delete, decline, we're not looking at your paper. What would be a better proposal to say, dear editor, we are aware of the many studies that have been published on this topic, where you mean, okay, this is an important field. Therefore, our manuscript really makes a difference because, and then you explain the reason, we are the first to demonstrate the mechanism. Given the attention to this topic in the scientific community, we believe that our manuscript could serve as a reference for many authors in this field. What this last sentence means, we believe that our manuscript could serve as a reference for many authors in the field, actually tells the editor, we think that this will push up the impact factor of your journal, because that's actually what it says. So that's how you could make a cover letter that could make the difference, where if an editor is in doubt and he reads your cover letter, he or she reads your cover letter, that they can be convinced to indeed go forward with reviewing your manuscript. After the review process, you get back. In the best case, they will say, okay, we can maybe accept your paper, but first you need to change these and these things. Therefore, you need to make a rebuttal letter, an answer to the reviewers and to the editor. It's very important that you take this very seriously and really respond to every comment that the reviewers and sometimes all the editors have made, one by one, very clearly, and make it as easy as possible for the reviewers to go through it. If you make it very complicated, then reviewers will be discouraged and will say, you know, we'll wait a bit, or we don't bother, we just do something else. If you make it very clear with color codes or even in a frame, that you can say, this was the comment, this is our answer, and this is how we corrected the manuscript at these lines, then it's very easy to look through that and you will have very good results. So indicate all the changes made, highlight them in the text. But also dare to disagree. If you really think and you're convinced that some of the comments of the reviewer, one of the reviewers, is not correct, then clearly reason about that. Then clearly say, I'm sorry, we accept that you make this comment, but because of this and this reason, we dare to say that it's not correct. So we keep what we have. So don't, you can disagree, but don't disagree because you do not like the comment. Sometimes you can have very harsh comments, but these harsh comments are correct because you might have done something wrong, and then accept the comment and dutifully try to change it. So some take-home messages here. Do not start doing research without knowing what has been done in the topic already. Literature review is really important here. Be systematic in data recording. Be critical about the data you record and how you interpret. Be honest. Don't try to don't try to be biased to what you would like to see. Be honest, because even if you find something you would not like to see, might in the end be something very interesting. So honesty is crucial in doing science, and keep it simple. It's not the most complicated studies that often are the best. It's usually the most easy ones, the most straightforward ones that give the best and strongest results. And with that, I would like to end my talk and open for questions.